Okay, good afternoon, everyone. Welcome to, sorry, good afternoon, everyone. Welcome to the Spring 2025 webinar series, which is uh, which is a thing that the Graduate College of Engineering (GSC) is doing every semester. Uh, so today we have Dr. Rupa Haldavnekar, uh, who is going to present her work to us. So before moving on to her talk, I would like to introduce the graduate student community at GCOE. So this is our team for the year 2024-2025. We have our two co-chairs, Rodney and Rachel. I think they are present here. Then Avinash and myself, we are part of the publicity team. So all the emails that you get regarding the GSC events that are coming from our side. Then we have treasurer Siavash, and then we have a pretty big support team. We have Mohammad Sarbaz, Louis, Roshan, Samuel, Jason, Himadri, Nurayan, and Mohammad Sadiqur. And then we have our mentor, Professor Farooq Mistry. So the GSC at GCOE leadership team meets every Friday at 4 p.m. The meetings are held online through Zoom. If anyone is interested in joining the leadership team, please contact Rachel or Rodney. Their emails are mentioned on the screen here. I'll give 5 seconds if you want to copy that, or you can just type Rachel or Rodney's name on the uh, Outlook search box, and then the email should pop up. So the meeting timings are flexible. If you cannot attend at 4 p.m. on Fridays, we'll try to adjust our meeting timings in case you are really interested into joining the GSC leadership team. Uh, I would encourage everyone to become the GSC GCOE member on Engage page. We have a QR code here that should direct you to uh, the Engage page of GSC. Please make sure you sign on to that page. Many of our events, you get the news through Engage as well. It is a good way to keep in contact with the GSC team. We also have a LinkedIn page. The QR code for that is given on this page. Like I said, so similar to Engage, we give our events information on LinkedIn as well, and then we also have some alumni part of the LinkedIn page where you can contact, you can get in connection with them and have a contact with them. We also have a GSC GCOE Facebook page. The QR code for that is here. You can scan the QR code, and it should redirect you to the Facebook page. You can find our event information on the Facebook page as well. Coming to the GSC GCOE YouTube page, you can scan the link here to go to the YouTube page directly. All our online webinars are posted on the page. If in case you miss some of the webinars a week or something, you can go to the YouTube page and get the entire webinar over there. So it is a good tool for you to have in case you in case you miss any session. So we have the GSC team has a Dave and Suzette Bird team room. This team room is available for all the GSC members. Sorry, just a second. The GSC team room, it has some good amenities like the couches, whiteboard, coffee pot, microwave. If you want to access this room, please email Rachel, Rodney, or even Dr. Mystery. We can have you access for the team. A quick reminder before we move on to the talk for today. In a few minutes, we will be putting the link for the Google form in the chat. Please make sure you fill out the seminar attendance form. It gives us a way to show for funding that we have people attending our webinar series. So please do not forget to fill in the seminar attendance. Okay, so moving on to today, we have Dr. Rupa Haldavnekar, who is going to talk about SERS ML or SERS ML liquid biopsy for cancer diagnosis. So Rupa Haldavnekar, she is an assistant professor in the School of Aerospace and Mechanical Engineering. Her research includes nanomaterial design, Raman spectroscopy, machine learning, and data analysis. She also has a research in oncology, especially in discovery of biomarkers for early cancer detection and development of point of care application. She has a PhD in biomedical engineering. So without wasting any further time, I would like to pass on the stage to Dr. Rupa and for presenting to us.
Thank you, Swapnil. I'm going to share my screen. Do you see my screen?
Yes, we can see.
Okay. So hello, everyone. Thank you, Swapnil, for the introduction. And thank you, everyone, for attending my talk. So today I'm going to talk about cancer diagnosis. This research is multidisciplinary and it integrates material science, physics, biology, and machine learning together. So the motivation behind this research is that the cancer burden has continued to grow globally, estimating around 20 million cancer cases and 10 million cancer deaths. One in every 6 people, every 6 deaths happen due to cancer, and more than 100 distinct diseases are encompassed by cancer, with distinct signature which characterizes uncontrolled cell growth, proliferation, and possibility of invasion, which is metastasis. So basically, we are talking about a disease which is created by our own cells, which is the source of this disease, which creates tumors. So detecting these cells, unlike detection of bacteria or viruses, is extremely difficult to do. And cancer is highly heterogeneous disease. So within a tumor, there are subpopulations of cells demonstrating distinct phenotypes, genotypes, and I'll talk about the heterogeneity later on. But this intra-tumor heterogeneity is instrumental for tumor evolution, and that is the main reason for treatment failure. So to understanding this tumor heterogeneity when you are detecting the disease is very important, because that is something, that information will give the oncologist to decide what is the treatment of action they are supposed to be taking. So I will briefly explain how currently the diagnosis is done. So before going into this detection process, first there will be symptoms which will appear. So a patient will start feeling nauseous, or they have headaches, they have some kind of symptoms which occur. And once the symptoms occur, the physician will do physical exam. So they will check for lumps or bumps that could suggest cancer, or they will look for skin discoloration, organ enlargement, that kind of analysis. Then they will send you to the lab to get your tests done. So typically urine blood test is done for any abnormalities. Then non-invasive imaging is done. So computed tomography, bone scan, magnetic resonance imaging, positron emission tomography. These are some techniques the lab will use to see if you have any presence of any tumors. And if they feel that there is some risk of tumor, then they will send you for biopsy. And tissue biopsy is done after imaging is done. And during that tissue biopsy, if they find any abnormalities in your histology, then they will confirmly say that, yes, you have cancer. And after that, they will start deciding on what kind of treatment you have to take. So tissue biopsy is the only way right now for definitive diagnosis of cancer. Now, let us see how this tissue biopsy looks like. So you see how the fine needle aspiration is done here, image-guided tissue biopsy is done. And you will see that all these tissue biopsies are so invasive, painful. And these are very, very invasive methods to detect cancer. Not only they are invasive, sometimes there is a huge problem doing tissue biopsy because sometimes you may not get enough sample from the biopsy to do your tests. So many of the tests cannot be performed. Or during early cancer, you cannot see tumor, so you cannot perform tissue biopsy. Then this is a highly unrepeatable process. Imagine doing this for a brain cancer patient. Tissue biopsy is not possible for any metastasis, which is the cancer which gets spread out throughout the body. At that time, that is not possible. And the most importantly, you cannot look at the tumor heterogeneity with tissue biopsy. So you are taking a slice of tumor from one location. You are just getting a snapshot at that specific instant. So repeated sampling and the tumor heterogeneity you cannot look at. So these are some of the limitations of tissue biopsy. Now, what I mean by heterogeneity is when there is a primary tumor and when the chemotherapy is given, the first line of treatment, the tumor changes its profile. Different types of cells from primary tumor to the residual tumor have changed now. So based on that, the second second line of treatment has to be done. So every time you have the second or third line of treatment, every time you have to go through this detection process to see what type of tumor you have now, so that the physician, the oncologist can make a decision on your treatment. So detecting cancer and identifying or monitoring these cancers, this is a very important area of biomedical engineering. Tumor heterogeneity is also spatial. So a tumor in brain does not look like a tumor in lung or a tumor in colon. And temporal heterogeneity, I just explained that after treatment, the tumor starts looking different. So all these aspects the oncologist has to keep in mind when they are trying to identify the best line of therapy for a patient. Typical tissue biopsy analysis is done using these histopathology, surface markers, mutation, gene expression, protein analysis. But these methods are very time consuming, they are very complex, and they are usually done in a late stage only after the symptoms start appearing. Then different types of imaging, these are non-invasive methods. So methods like MRI, ultrasound, PET scan. PET scan is pretty good option, but PET scan uses radioactive agents. So it is a very detrimental for, sometimes detrimental for patients when PET scan is done on them. You are getting exposed to radioactive agents. And the limitations of imaging are that you can do structural investigation, but you cannot identify what grade or what subtype of tumor you have. Or you cannot tell apart from a tumor, which is a recurrent tumor versus a residual tumor. Sometimes the scar tissue, which is created due to chemotherapy, also gets diagnosed as a tumor. So that is a huge false positive. Also, imaging gives you a snapshot, like a static snapshot of this ever-changing or ever-evolving tumor. So all these limitations of current diagnostic are making the field go towards liquid biopsy. These are different types of methods for imaging, and I have highlighted the limitations of these methods. One of the most important limitation for PET scan, which is a very expensive process.
بحثش میدیم یه ریسرچ اینترست تو بگو.
Sorry?
بعد به میگه علی.
It's okay. Yeah, so PET scan uses radioactive agents, and usually they use fluorine-18 as their radioactive agent. The half-life of fluorine-18 is 110 minutes. That means not even 2 hours. So you need to use that manufactured fluorine-18 right away when you want to do this type of analysis. So rapid synthesis, transportation, and imaging before the decay of that fluorine-18 happens. So these kind of limitations all these imaging techniques have. So diagnostic field is moving towards liquid biopsy. Now, what is liquid biopsy? It is a non-invasive reproducible sampling method. You are taking some blood from the patient and you are trying to identify presence of these tumor-related biomarkers in your body fluids. This method has potential to capture tumor heterogeneity because now the biomarkers from various locations of tumor are getting secreted in blood. It's possible to analyze multiple biomarkers. It's not like you are just identifying one mutation or you are trying to identify one specific protein expression. You are trying to identify multiple biomarkers. This method does not have any side effects. You are not getting exposed to any radioactive agents. And this can be used for multiple applications like disease diagnosis, treatment monitoring, prognosis of treatment, that kind of applications. So typically for liquid biopsy, circulating biomarkers are used like upregulated proteins or peptides or DNA secreted from tumor, which is called a circulating tumor DNA, extracellular vesicles, or tumor cells. Tumor cells which are detached from the tumor are very rare to find in liquid biopsy. So even in late stage cancer, it's very difficult to identify those. So other markers are typically used like circulating tumor DNA. Many of the liquid biopsies use that. But and you can pretty much identify it in stage 3 or stage 4 patients. This market share of liquid biopsy technology is growing exponentially, but currently very few tests have been approved by FDA. And you can see from this table that these tests are mostly for one single cancer, and they either have very low sensitivity or low specificity. So use of these tests currently is only limited as a companion diagnostic test. And there are few challenges for current liquid biopsy, and I like to categorize those into 2 categories. One is a technological challenge, which is due to the current technology, and one is a biological challenge, which is due to the challenges of biomarkers for cancer. So limitation of current technology, if we look at, they need very, very high purity of sample. Then they also need high yield of these biomarkers from very low biofluids. That means you need very, very high blood volume. Sometimes they will also ask for 10 milliliters or 20 milliliters of blood, which is very difficult to get from a cancer patient. Then it's not feasible to perform concurrent analysis of multiple markers simultaneously. So you can either detect one protein expression or one mutation. Detecting an array of biomarkers simultaneously to give a conclusive result is not possible with these liquid biopsies. And trace level biomarkers at early stage are almost invisible for these liquid biopsies. The biological challenges are the biological biomarkers are many times non-specific to cancer. For example, if we are looking at a certain mutation like TP53 mutation, it's present in ovarian cancer, esophageal cancer, colorectal, head and neck, lung cancer. So you can say that there is an elevated level or there is a TP53 mutation present, but you cannot tell which location this cancer is present. That means the oncologist does not know how to treat that cancer because what you need to know the location of that cancer to treat it first. Then these biomarkers have very short half-life, 2 or 3 hours. So they get easily degraded in blood. So also during the purification, isolation processes, the integrity of these biomarkers get badly affected. So these liquid biopsy tests have become like a yes or no test. And it's very difficult to identify the tissue of origin using these tests. So when I started my research, I had few things to keep in my mind. These are the design considerations I want to keep in mind. I want to identify cancer-specific biomarkers. That means if any asymptomatic individual comes in for a liquid biopsy test, we should be able to tell them if they have cancer or no. If we suspect cancer, then what is the likely location of that cancer? Is it a likely location is breast or lung? If we identify that suspected location is breast cancer, can we also tell them what type of breast cancer it is? Is it HER2 positive cancer? Is it a triple negative cancer? Does it have any metastatic potential? So all these things, if we are able to tell in a liquid biopsy test, then the clinicians can decide the line of therapy for these individuals. So my aims are to identify such biomarkers, which are cancer specific, to develop a test which can identify these rare biomarkers. That means trace level detection. So we need ultrasensitive detection. This should not need a lot of sample prep because during the sample prep, we tend to lose a lot of material. So we don't want to lose important information from this sample. So not a lot of sample preparation is required. And we should be able to analyze multiple signals simultaneously. That means this test should also be multiplex test. And that will enable us to identify tumor heterogeneity. So these are my design considerations when I started my research. And I use Raman spectroscopy as my detection technology because Raman spectroscopy is a versatile tool. I can use a benchtop Raman microspectroscope for imaging, biomarker discovery, and if I need to, I can also use a portable Raman spectroscope for rapid point-of-care diagnosis. So there is a versatility with this tool. Now, let me just explain briefly what Raman scattering or what I mean by I'm using Raman spectroscopy. So Raman scattering is the inelastic scattering of photons by matter. When light is irradiated on molecules, the molecules scatter light. And Raman effect is based on a shift in the energy of the scattered light. So now we are also integrating physics in this research. This energy shift provides a unique molecular fingerprint reflecting vibrational, rotational, and low frequency modes of the molecules. Basically, we are getting information on a molecule in a form of a cryptogram. So Raman effect can be used to obtain fundamental information on biomolecules. This is a spectrum from a cell. Cell produces finger-like spectrum, fingerprint-like spectrum with unique detectable contributions from abundant cellular macromolecules. So each peak in this spectrum means something. For example, this peak here this is the peak for nucleic acid. This peak here, this is for a phenylalanine protein. So we use these peak positions, peak intensities, area under the curve, peak shifts, the shoulders of the peaks, like the shoulder of the peak here, as our information base for our analysis. But Raman signal is very weak. So we need to improve this enhancement, or we need to enhance this signal, which for which I design nanostructures, which gives general signal enhancement. So now we are also integrating nanotechnology in our research. So one mechanism is of localized surface plasmon resonance, which is electromagnetic enhancement. Another component is chemical enhancement or charge transfer mechanism. Because nanoparticles increase surface area, there are more molecules which can adhere to the surface, so the signal improves. These are some transmission electron microscope images and scanning electron microscope images of the sensors which I have designed. This is a nickel oxide nanoprobe platform. If you look at the scale here, this is 20 nanometers. So you will see that all these nanoparticles are extremely small. And because of the small size of these nanoparticles, the material starts changing its properties. And we use those changed properties for our signal enhancement. This SCM image you see here, it shows the network of all these nanoparticles, and it creates these small little voids in this network. So these voids provide a very good molecule trapping mechanism. Then there are multiple layers of these voids which act as a photon trapping mechanism, which results in photo-induced charge transfer. And the probes themselves have very large surface area. And nickel with cubical shape particles create surface plasmon resonance. And therefore, we observe orders of magnitude of signal enhancement. So this is a FDTD simulation of a nickel nanoparticle. You will see at the corners of this cubical shaped nanoparticle, you see substantially enhanced surface plasmon. And when this nickel is coupled with nickel oxide, you get a metal enhanced semiconductor charge transfer mechanism. This is the simulation of two nanoparticles which are together, and we see enhancement improves by multifold. I also tried to use gold nanoparticles to decorate on this nickel oxide nano platform. That improves the signal much more. Then I also tried using graphene quantum dots, zinc oxide, zinc nanoparticles. So all these nanoparticles which have semiconductor-based oxides, I tried to use to see if I get the metal-enhanced charge transfer mechanism in that. So this is the spectra of crystal violet. This black line shows you that crystal violet does not have any nanoparticles with it. The pink line shows you large nanoparticles. You see here 50 nanometers. And the purple line shows you the platform which I just showed you before, which is the nanostructures which are 10 nanometer small. So you will see that the signal is enhancing so substantially, and it also improves the sensitivity of the signal. And because it is improving the sensitivity, we also can look at different peaks which we will otherwise not see without any enhancement. And this helps us, this is the crux of all the ultrasensitive sensing. And this is what gives us trace level detection and also more sensitive signal. This is a schematic of a cell with this nanostructured platform, and you see different peaks which belong to nucleic acids, lipids, proteins using these nanosensors. If these nanosensors were not there, you will just see this black flat line here, and you cannot make anything out of it. So this is this is the technology behind my research, and this is how the raw spectra looks like. So looking at the complexity, and because this is label free, we need to apply analytics on it, which should be advanced analytics. So we use unsupervised multivariate analysis, like principal component analysis. And we also once we explore this data set, so principal component analysis is typically exploratory analysis, and we try to get much information from it, which peaks are responsible for giving us some information. We pay a lot of attention to principal component loadings and principal component scores. That way we can like filter out the significant peaks. And once we extract those peaks or features, then we can apply supervised machine learning. And I typically use artificial neural network and partial least square discriminant analysis to analyze this type of data sets. So this is a green spectra, which belongs to a non-cancer cell line. This blue and pink spectra belong to cancer cell lines. So blue spectra is for a triple negative breast cancer. Pink spectra is for cervical cancer. And if you look at this principal component analysis, you see 3 different clusters. That means we are not only detecting presence of cancer, we can also tell you what type of cancer you have. So that is very much possible using machine learning. And we also rely a lot on ratio analysis when we are doing analysis using spectroscopy. So this ratio is typically the ratio between proteins to lipids in the cells. Cancer cells tend to get more stiff, so more lipids are present in cancer cells as compared to normal cells. And that gives us this less protein to lipid ratio for cancer cells versus high protein to lipid ratio for non-cancer cells. So once I was satisfied using these sensors on cells, then I started thinking about applying it on different biomarkers which we use in liquid biopsy. So one hallmark of cancer is the aberrant changes in the DNA structure. So I wanted to use the structure of DNA as one of my biomarkers. So detecting circulating DNA at early stage, we are using these ultrasensitive sensors. So this sensor can detect up to femtomolar concentration of DNA. That means in terms of early cancer diagnosis, brain cancer secretes the least quantity of DNA, which is like 0.1 nanogram per milliliter to few nanograms of circulating tumor DNA. Current liquid biopsy needs 1 milligram per milliliter of DNA. So brain cancer is invisible to current liquid biopsy during early stages. But my sensors are able to give limit of detection up to 10 power negative 4 nanograms per milliliter. That means this can detect early cancers. You can clearly see the bands for adenine, cytosine, guanine, and thymine here. So these are the ACTG molecules which make, or nucleotides which make the DNA. And using these structures, we can try to identify presence of cancer. And this is the method which I used in my previous studies. So this idea is to generate training data from isolated tumor DNA, isolated cancer cells, as well as cancer stem cells. I use those to isolate the DNA. And therefore, now I have the training data from the cancer cell DNA and cancer stem cell DNA. And then we want to incorporate tumor heterogeneity by using cancer stem cell DNA because cancer stem cells are the originators of tumor. And then I also compared that spectra with the circulating cell-free DNA because the circulating cell-free DNA has cancer DNA as well as the DNA from other cells which are non-cancerous cells. So you want to identify those specific peaks which belong to the tumor DNA. So once we have that done and once we have that training data, I simply used patient plasma as my testing method. And we got pretty good sensitivity, specificity for identifying presence of cancer. But identifying tissue of origin with this method was not very accurate. So I wanted to also incorporate not only DNA, but proteins and lipids also as part of this analysis. So what is the best way of doing that? So I started thinking about other biomarkers, and then I realized that if I use extracellular vesicles have secreted DNA, RNA, proteins, and lipids in it. It all actually gives you like a mini map of a cell. So if we can use extracellular vesicles as a biomarker, then we will probably be able to get more accurate diagnosis as compared to the diagnosis using just the DNA. Very interestingly, the extracellular vesicle cargo of cancer cell looks very different from that of cancer stem cell. So you will see here not only the spectra look different, but if you look at the heat map of the significant peaks, this is for lung cancer cell and lung cancer stem cell. So the cargo looks very different for these two types of cells. So if we can combine this data, we can also incorporate tumor heterogeneity, and we can try to find out if this is a good biomarker for cancer diagnosis or not. What we observed was cancer stem cell extracellular vesicle show unique profile as compared to the non-cancer extracellular vesicle. With just the cancer cell extracellular vesicles, there is an overlap. In the dendrogram here also, you will see there is an overlap. So we decided to use cancer stem cell extracellular vesicle as our biomarker. And we were able to do very good diagnosis for brain cancers. This specific one is for glioblastoma. So you can see clustering here. You see significance in different peaks. You also see different spectra. So these purple spectra are the spectra of glioblastoma cell lines, and the black spectra is so these are cancer stem cells of glioblastoma cell lines, and these are the these are the neural stem cells. So you see variation in the spectra here, and using this type of information, it was we actually got very good results. So I find extracellular vesicles a better biomarker than just using circulating tumor DNA. Going forward, this biomarker discovery is my ongoing research. So last few years, I have been using immune cells for cancer diagnosis. Now, why do I want to use immune cells? Is because it's the first to respond to the presence of tumors and viruses. Immune cells are recruited by tumor cells, which is the main factor which causes immune cells to accumulate in tumor microenvironment. And the prognosis for cancer, the response to treatment are correlated to the density of immune cells, the function of immune cell population in tumor. So immune cells interaction with tumor can give us a lot of information. And immune cells are also pro-tumor and anti-tumor. So my hypothesis is that during the first few stages, like early stages of cancer, immune cells will be anti-tumor, and then they convert to pro-tumor phenotype. So using those functions of these immune cells, we should be able to identify whether the cancer is in early stage or late stage, what type of cancer it is. And if you look at this diagram here, every immune cell has different type of spectra. So T cell has a different spectra than natural killer cell or monocyte. So if we take each type of immune cell separately and then combine all those as a biomarker, that will give us very accurate diagnosis. So I started this research using natural killer cells because natural killer cell is the first line of defense. They don't need any molecular signaling to get activated. So we did an experiment. We activated these cells using cancer stem cells, and we observed that tumor-interacted NK cells show distinct profile as compared to the uninteracted NK cell. So here you will see that this purple spectra, this purple cluster is for the breast cancer stem cell associated NK cell. And this is the spectra of brain cancer cell associated NK cell. So the NK cells also have different type of activation mechanism around cancer cells versus cancer stem cells. They recognize the cells in tumor separately, and then they can switch to an active mode. We see intracellular changes in NK cells. We see that there is increased stretching of disulfide protein bands. There is decreased tyrosine, phenylalanine. There is decreased nucleic acids in these NK cells. And then we use these models to train our algorithm for cancer diagnosis. This is showing for lung cancer and colorectal cancer. So for different types of cancers, we still see similar trends. And one observation was that NK cells interacting with tumor did not show very homogeneous signals. Rather, they are unique and heterogeneous with distinct subsets. So principal component analysis did not demonstrate clear clustering between cancer and non-cancer cells, which are associated with NK cells. And this overlapping clustering can possibly because immune escape phenomena is exhibited by cancer cells, which are interacting with NK cells. But NK cells are preferentially cytotoxic to cancer stem cells. So my hypothesis is that training machine learning algorithm with the signals of NK cells interacting with cancer stem cells will be a better training data set for diagnostic accuracy. So we did experiment to confirm this hypothesis, and classification was very good. Keeping in mind that we have a small data set here, so to conclusively say that this is the best way of diagnosis, we need to do a large scale study or even a clinical trial for that matter. Tissue of origin detection also showed good accuracy as compared to the DNA structure where we were not able to classify between breast cancer versus colorectal cancer. Here we can get good accuracy, good specificity for the classification. This specificity is reduced here between breast and lung cancer, and I'm trying to figure out why do we see similar kind of response from NK cells for breast and lung cancer. So that is ongoing research right now. We also did some analysis on different types of algorithms, just to see that is this, whatever we are doing, is this diagnostics algorithm specifically? That means, am I able to get these good results because I'm using a certain type of algorithm? But that was not the case. So here you see PLSDA analysis and ANN analysis, and the results are pretty much the same. So then we were able to say that our diagnosis is not algorithm specific. We are actually identifying good biomarkers to do this kind of diagnostics. So going forward, now my plan is to combine the results from all three biomarkers together. That way we are getting data from tumor using the structure of DNA and the extracellular vesicles. And we are also getting data from immune cells. That means we are trying to create the information on the tumor immune microenvironment. And I'm hoping to identify new biomolecules that will be instrumental in finding immunotherapeutic cures for hard to detect cancers and treat those cancers. I'm also exploring the advanced imaging modality where we perform SERS imaging and also use data from individual spectra. So this is the next project, and we will use SERS imaging and multimodal machine learning approach for early detection of cancer. Machine learning will help us with discovery of unexpected biomarkers with potential for therapeutic intervention. And for this, we are also in a process of developing lab-on-a-chip platform for simplifying sample handling and standardization of sample storage so that this research can be translated from lab to the market. That's it from me. And thank you so much for listening to this talk.
Thank you, Dr. Haldavnekar, for presenting your work to us. With that, I open the platform for any questions that you might have for Dr. Haldavnekar. Yes, Roshan, go ahead.
Oh, sorry.
No, Rachel, you go ahead.
No, just a couple of quick questions, just because I've got another meeting a little after 1. So one quick question, you said you had like a small sample size for that last experiment. Like how many people were you able to test that on?
We had 60 samples. So we had 20 samples of glioblastoma, 20 samples of astrocytoma, and 20 samples of oligodendroglioma. So high-grade brain tumors versus low-grade brain tumors. And then we had 50 samples of non-cancer, like 50 blood samples of non-cancer patients. So non-cancer, healthy donors. So that was the sample size. But considering this, to do the robust analysis, I feel like at least 50 to 60 tumors per sample, brain, like per tumor type should be a good sample size going forward. Considering rarity of this disease also, it's not very easy to get the tumors. So
Yeah, that's always the
glioblastoma is
problem
like
with medical data.
15,000 patients in a year. It's all over the world. So it's a rare disease. So getting those samples is also difficult.
Yeah,
We are
understandable. I feel
actually
the
writing
exact
an
same way.
IRB with OUHSC right now to have a blood, like a tumor bank in OUHSC, so we can get more samples from there.
Rad. I guess other question, you said you were going to kind of look at using machine learning to find like, unknown patterns. Are you planning on using like specific unsupervised techniques or?
So we are going to use long short memory convolution neural network for that. And we also want to do multimodal analysis. So we have this data from Raman data sets from like DNA and extracellular vesicles is our tumor data. Immune cells is our crosstalk of tumor with immune environment. So it becomes like two different data sets. And then we also want to combine the clinical data with that so that we should be able to identify some patterns out of it.
Cool, very cool. I will let others ask questions for it.
I just wanted to know about your training data, because I'm assuming that some of these tumors, how they develop or how their characteristics are, may change with like changing medication and things like that, and how they react. And so I don't know, like, how do you account for those sort of changes? And let's say 2 years ago, maybe there were things that you could like test, and now with the improvement in medical science itself, like, how do you plan on like improving on your testing data so that the model itself is sort of up to date with the current testing
Yeah.
methods?
So the model will keep on improving, like
It's
improving
just
over the period of time. But
okay.
we are trying to get this longitudinal data sets. It's not very easy to get longitudinal data, like historical data
Right.
for samples like tumors, especially because
Right.
they do biopsy once and that's it, right?
Right.
So blood samples is a better option to
Right.
get the data from the patients. And right now I'm working with somebody at OUHSC to get ascite samples for ovarian cancer patients. So these ascites, they time and again they try to get extract like this. These are like small little pockets which happen in ovarian cancer patients. So they have to extract them over a period of time after every treatment. So that gives us like the data set, which is like time resolved data set, and then we can look at it to see if we can find any patterns in it.
And I'm assuming most of the markers that you're checking is specifically for that tumor, right? Like you need that tumor to test. Is there any way where it's like even less invasive, in the sense that you use blood as the sample and then use some of these markers instead of having to use the tumor in itself, and that place you could just draw some blood and then like look through the
So
model?
that will happen eventually
Okay,
because
right.
we need something to train our model on.
Right,
So
right.
the initial model has to be trained on tumor. Otherwise, we will not know what markers are there in the tumor.
I see.
But once we have tumor and we also have blood from the same patient, we try to see the correlation between those spectra.
Right.
So once we have that information, then the information from the blood can only be used as training data.
I got
But
it.
that will happen over a period of time, not right away.
Okay, sure. Thank you.
Do we have any more questions? Yes, Dr. Naidu.
Yeah. So just I would like to comment that this is a very interesting field, and I was working in Human Genome Project, so DNA spotting robotic system we were developing. So pretty long back, but thank you very much, Rupa, for a very nice presentation, very detailed. And I think this has very great promise for diagnostics, because it can solve this taking the tissue biopsy. And I think it's very detailed and very comprehensive work. Please go ahead, and wish you all the best for your research. Thank you.
Thank you so much, Dr. Naidu.
Okay, so do we have any more questions? Rachel, did you have any other questions?
A few. I'd kind of like to look at the papers as well a little bit. And I guess like you said you used neural networks for that first work. What kind of neural network were you using?
It was artificial neural network.
Just like a
It's not a
feed-forward one?
very, sorry?
Just like a feed-forward neural network?
Feed-forward neural network, and it's not a very complex algorithm. It was a very simple one. The whole idea behind keeping it simple that way is so that I don't want my diagnostics to be based on the neural network. I mean, it doesn't have to be really, really good results because it's the algorithm which is giving me that. And then I don't know why that is happening. So that's why I'm more partial to PLSDA analysis also, because that gives me latent variables. That also gives me very important peaks in my algorithm. And it also tells me which specific peak has what percentage of importance in the decision making. So you looking at those peaks, I can again go back to the biomolecular composition of cells to see what exactly is happening in the disease. So I don't want to use like very complicated black box type of networks.
Yeah.
Okay, do we have any other questions? Okay, so I would like Dr. Mistry to speak a few words. You are on mute, Farooq.
Rupa, thank you very, very much for this presentation. It was beautiful. The diagrams and the pictures that you had were absolutely gorgeous. And I know nothing about the subject, and I'm intrigued because cancer is a very important. I mean, cancer causes a lot of deaths, and anything that could help in terms of mitigating it would be very nice. And I'm really happy to see that you're interested in this and that Queenie is working with you, and Rachel had all these lovely questions to ask you about what's going on. And it's my hope that people will get in touch with you so you'll be able to form a small community to take it further. And it's
I'm
really
hoping
to
so
have
too.
Trenton, the undergraduate, with you to attend, and maybe he can get other undergraduates to work with you on this very interesting topic. So that's
Thank
my
you
two
so
penny
much.
bit. So
Thank you. Really appreciate it.
So as it is our practice, I'm going to stop recording, and then we can go into the chit chat mode and talk about